So anytime I refer to phenotype, I'm talking about physical traits that you can see with the naked eye. Whenever we talk about genotype, we're simply talking about the genetic code or the genetic type. The organism's phenotype is tied to the genotype. The genes are going to control all functions of a host cell. So genotype and phenotype are pretty much indicative of each other. It's just that with phenotype you can visually see the outcome of what the genotype is. And so when you think of a genetic code, you think of things like A, T, G, C, and then their complementary base pairs. So that would represent what the genes would be. And when an organism is alive and expresses those genes, then traits will be expressed visually so that we're able to see them. And then we refer to that as the phenotype. So an example of a phenotype would be like a seed that is going to produce purple flowers. Okay, and Mendel's law of inheritance, he basically studied flowers and looked at the variation of colors that were produced when certain parents would crossbred with each other. Okay, but essentially that's only made possible through the process of genes, and genes are a gene is defined as a segment of a chromosome. Okay, so let me blow up this image here because I think it's too small for you guys to be able to see. In this image here we have an image of a small little critter, and the small little critter has a specific genotype pattern here that's embedded inside of these chromosomes. And what these chromosomes are going to be coding for is essentially some attribute for this particular organism. So without those particular genes or genotype, this organism isn't going to express the gene. Okay. So genetics is very interesting, especially bacterial genetics, because this is all bacteria are little clones of each other based on the way that they're replicated. Our animals, in typical, they don't produce via the process of essentially what we refer to as cloning, which is also known as binary fission, where you have one cell splitting directly apart into two identical cells. Our cells don't do that. There's a lot more variety and a lot more of exchange of chromosomes. And when we talk about exchange of chromosomes, we're talking about exchanging pieces of genetic material from, say, this chromosome to this chromosome, which gives us variation in the genetic code. Now, changes in the genetic code are referred to as alterations, and these alterations are better known as mutations. So mutations are pretty much always going to be possible whenever you have replication of DNA, or you have DNA being passed from parent to offspring. There's three types of mutations. There's those that are harmful, there's those that are beneficial, and there's those that are silent, which means they're not really harmful or beneficial. So mutations are very interesting to us because they can occur inside of the genetic code, and when they occur, they could lead to some massive changes in the organism itself. In some instances. In other instances, maybe the mutation just doesn't happen and doesn't really make all that difference in terms of expressing certain traits for a specific organism. But if an organism does undergo a mutation in its genetic code, we refer to that organism now as a mutant. And there's a lot of factors that are involved in causing mutations. One such factor is exposure to environmental hazards, so things like UV light from the sun that can alter the genetic material because it causes the hydrogen bonds between the nucleic acids making up DNA, such as adenine, adenine, thymine, guanine, and cytosine to basically kneel with each other, or it can produce what's called these thymers. And so if you have too much exposure to UV light, that can generate cancer, which is a byproduct of mutations that occur in the genetic code. And when you have mutations, the codes aren't read properly. And remember, in the central dogma theory, DNA is transcribed into messenger RNA and then translated into protein. If DNA is not transcribed correctly and RNA is not translated correctly, then you're not going to have the correct production of a protein, which is going to be indicative of a mutation that is either going to be observed in a phenotypic fashion, which means you can see with the naked eye, or it'll be in the genetic code and be expressed eventually later on in life. So here are some effects of mutations. This primarily relates to those of bacteria. And the best example that we have in bacteria regarding mutations is that of antibiotic resistance. Okay. There are some antibiotics out there that use a specific molecular structure that's known as a beta-lactam ring structure, and that's what this molecule is right here. And this beta-lactam ring structure is used to break apart or drill a hole, essentially, through specific bacteria, primarily bacteria that have very thick layers of peptidoglycan surrounding their cells. So beta-lactam ring structure will drill its way into a normal bacterial cell. And as it's doing that, it punctures the cell membrane and enables the cell to essentially succumb to being overrun by any solutes or any solutions that are outside of the cell, because they'll rush into the cell and then they will cause the cell to what we refer to as lyse. Now, if you have a bacteria that has a mutation inhibiting this beta-lactam structure from operating on the peptidoglycan, then we start to get resistant bacteria. So you have bacteria that can be resistant to multi-drugs, kanamycin resistant, amoxicillin resistant, novomycin resistance. The best one is methicillin resistance, which some members of Staph aureus, the Staph aureus organism, are a part of. So that resistance basically just means that all the bacterial cells have some specialized mutation which allows them to survive being attacked by or trying to be destroyed by a specific antibiotic. And in this instance, many of those has to do with the beta-lactam ring structure, which is found inside of an antibiotic. So think of something like penicillin, and it's used to drill a hole through the bacterial cell, so that way the bacterial cell will lyse and die. But that doesn't happen if you have the resistance gene being produced in the bacteria. Okay, so let's talk a little bit more about how bacteria take on new genetic information. Like, how does a bacterial cell that was once impacted by an antibiotic like methicillin go from being subjected to an antibiotic to being resistant against the antibiotic? Well, there's a couple of ways this can happen. And so we have essentially the processes of conjugation, transformation, transduction, and then lysogenic conversion. Now, in the process of lysogenic conversion, this actually requires the use of what we refer to as a bacteriophage. And a bacteriophage is a virus that contains DNA or RNA, and that phage is going to inject its DNA into the bacterial cell, such as a syringe would inject an antibiotic or a vaccine into a patient. With the injection of this DNA, the bacteriophage's DNA is going to integrate itself into the DNA of the bacteria. And bacteria, if you don't know, they have DNA that's in a circular formation. So once the bacteriophage has integrated its DNA in the recipient bacterial cell, that bacterial cell now contains recombinant DNA. So that's one way that a bacterial cell can acquire new DNA or new genes. Transduction is the process where you still have a bacteriophage being used, and a bacteriophage is going to, again, inject its DNA into the cell, and once it injects itself into the cell will start to integrate the bacteriophage DNA into its bacterial chromosome. The bacterial phages or the bacteriophage's DNA will start to fragment the DNA of the bacterial cell into small little phage replicates. And then those replicates will package themselves in the form of bacteriophage heads, which are going to be viruses. And this becomes lytic, a lytic process for the cell, because then the cell gets inundated with these bacteriophages carrying its DNA, and that starts to cause the cell to burst or to lyse. So then all of these small bacteriophages are just released into the environment, and they go and they infect another bacterial cell to integrate their DNA into them. So that's what happens in the process of transduction, is a simple integration of DNA from a bacterial phage. And a bacterial phage looks kind of like a little syringe. It has these long legs on it. It has a viral capsid at the top, and it will adhere to the membrane of a bacterial cell and then inject its DNA into the cell. In the process of transformation, this is where a bacterial cell simply comes into contact with naked DNA surrounding its cell membrane, and then the cell membrane will allow the DNA to pass into it, and once the DNA gets into the bacterial cell will just start to transcribe that DNA and then translate it into a protein to express it. In the process of conjugation, you actually essentially have two bacterial cells that will pass DNA between each other through the connection of a structure known as a sex pilus. And so one donor bacterial cell will transfer new genes into a recipient bacterial cell, and then that recipient bacterial cell will be able to express those genes once it replicates them. In a bacterial cell, they have specialized structures that can pass through something like the sex pilus, and these are called plasmids. So I'm sure you've heard somewhere in your career something called plasmid DNA. All that means is that you have a specific DNA that could code for something like resistance in a bacterial cell. And that resistance gene is going to be in circular formation. It's a small segment of DNA, but it is in circular orientation, and that can pass through a sex pilus and go from a recipient cell or from a donor cell to a recipient cell to convert the recipient cell into a resistant bacterial cell. So that's an example of an organism or bacterial cell as it acquires a new plasmid or a new gene. And here's kind of an image of some chromosomes. This is a image from your book where you see that we have an E. coli cell where DNA has essentially been spilling out of the cell. So all of this is DNA. And then right where this arrow is, we have a plasmid that you can see. The plasmid is a circular DNA component. Bacteria only have circular DNA, and their DNA is not like ours. It's not double helix. It's just circular in fashion, or it's not double helix linear DNA. It's circular DNA. And here's an image of that plasmid. And plasmid is what would code for a resistance gene, possibly. All of this DNA out here would code for all of the other structures that this bacterial cell needs to survive. So that's what the genetic material of a bacterial cell looks like. And in the processes of the lysogenic conversion and also lysogeny, you essentially have these bacteriophages, like I said, which are used to inject their DNA into the bacterial cell. Once that DNA is in the cell, the DNA will integrate with the chromosome, so this chromosome here. And once integration occurs, if this does not result in the death of the cell, then you have a process known as lysogeny, where your phage DNA is integrated into bacterial DNA.